take up.
This lecture is going to be on the organelles of the cell, and we're first going to start with the mitochondria. So if you look at this organelle, right here, so the organelle, the mitochondria. The mitochondria, and you can see that it's made up of an outer membrane, inner membrane, and within the inner membrane, you have this area that's referred to as the matrix. There's liquids in there that has chemicals that allow for parts or cell respiration to occur. The mitochondria also has a chromosome, so it has its own DNA. So it can actually reproduce on its own. It doesn't need the DNA from the cell, for the most part, for it to reproduce. And the number of mitochondria will change depending on the energy demands of the cell. So if the cell requires more energy, more mitochondria can be produced. And the mitochondria itself is the site of aerobic cell respiration. So aerobic means oxygen. So it's the part of cell respiration that requires oxygen. And this is where most of the ATP of the cell is produced. When there's not enough oxygen, the cell will produce ATP without oxygen or anaerobic respiration. And that will not take place in the mitochondria, but it occurs within the cytoplasm of the cell, the cytosol specifically. Now, ATP, adenosine triphosphate, this is often called the energy currency of the cell, but it's the energy molecule of the cell. And ATP is used as the energy source for any process in the cell that requires energy. The mitochondria, if you look at it, its numbers, we said, can vary. And they do actually wear out. So after a while, the mitochondria is replaced with new mitochondria in the cell. Now the ribosome. So if you look down here, the ribosome, this is an organelle that doesn't have a membrane around it. It's actually made up of two parts. And the ribosome, this is the organelle that acts as the site of protein synthesis or the production of proteins. So the ribosomes will read messenger RNA, and they will assemble the amino acids that produce proteins. They can be found just floating around in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. Now, down here at the bottom of the page, I drew a picture here. This part here, this is the cell membrane, and these little blue hair-like structures from there, that's cilia. So they're often described as thin fibrous structures that move back and forth. And when they do that, they move liquid across the surface of the cell. And they can be found on cells that line cavities in the human body, such as that's found in your respiratory system. And in the respiratory system, what they do is they actually will beat back and forth and move mucus out of the lungs to the back of your throat where you swallow it. But in addition, you can find them on cells, independent cells. Within the human body, there's no independent cells that have cilia on it. However, you'll find it in single-cell organisms that live in, for instance, water, that will have cilia, and they use it for locomotion. So in humans, cilia is only found on cells that are moving fluids across the surface, such as mucus, but none on independent cells. Now, the next organelle is the Golgi apparatus, and this is really another fascinating organelle. It's made up of a stack of these membranous little sacs, and you can see on this one here there's 1, 2, 3, 4, 5 of these membrane sacs, but they vary. You could have as few as like 3 or 4 of these, or you could have many of them. And this is the organelle that's responsible for modification, sorting, and packaging of chemicals. So if you see these little round structures here. Those are chemicals, bags or membrane bags of chemicals that are. This one here is being probably received from an endoplasmic reticulum, and it could be from the smooth or rough endoplasmic reticulum, and then it's passed from one membrane to another membrane, and it'll go through the series of membranes, and each time it goes through a different membrane, the chemicals can be modified. And then they'll be sorted, and then they'll be packed up, and either they can be packaged and exported from the cell, or they may end up staying within the cell. So it's kind of interesting because the Golgi apparatus can receive chemicals from both the rough and the smooth endoplasmic reticulum. And as the chemicals are being produced, they have a indication of their destination, so they can be produced and packaged up in the Golgi apparatus, and go for a specific destination within the cell or outside the cell. Now, if you look over here, you'll see the smooth endoplasmic reticulum and the rough endoplasmic reticulum. What's the difference between the two of them? Well, the smooth endoplasmic reticulum does not have ribosomes on it, where the rough does. And the ribosomes are the site of protein synthesis. So we can probably gather from that the rough endoplasmic reticulum is where you're going to have proteins being produced. The ribosomes are on the outside of the endoplasmic reticulum. And then what happens is a messenger RNA is read by the ribosome, and the protein goes inside of the endoplasmic reticulum, and that's called the lumen, the inside. And then it can be then packaged up and sent to the Golgi apparatus. The smooth endoplasmic reticulum does not have ribosomes on its surface, so its function is different. You'll find this is the site where you have not proteins, but lipids that are synthesized. And these organelles, the endoplasmic reticulum, also act as a transport system within the cell. And the smooth endoplasmic reticulum can act to detoxify chemicals. And if you notice, you'll see here, I actually drew the nucleus. So the ER, the endoplasmic reticulum, both the smooth and rough, is actually continuous with the nuclear envelope of the nucleus. All right, and then if you look at this bottom picture here, what I did is I drew a section of a rough ER, endoplasmic reticulum, where the ribosome is attached to the outside of it. And you can see there's these looks like a little tail coming off them. That's actually a protein that's being produced. So you'd actually have, and I'll draw it in blue here, you would have a messenger RNA that's being read, and as it's being read, that protein is being produced. All right, and they'll go inside the lumen of the rough ER. Now the nucleus. So up here in the upper left-hand side, I drew the nucleus. There's a nuclear envelope that surrounds it. So this is the nuclear envelope. Okay, and that nuclear envelope, you'll see spaces in it. Those are called the nuclear pore. And within the nucleus, you'll see these chromosomes. Chromosomes are only visible during cell reproduction, so during mitosis. Normally, they're not visible. And they're made up of, you'll find DNA that's packaged in chromatin within those chromosomes. And then within the center of the nucleus is a nucleolus. So the nucleus contains the genetic material of the cell. The nucleolus is the site within the nucleus where ribosomes are assembled. It's a location. There's no membrane around or anything like that. It's actually part of the nucleus. And then here we have the flagella. So you'll see a flagella here, and the flagella it's a whip-like structure. It allows for locomotion of independent cells. In human beings, the only cells that have flagella are male sperm. And then down the bottom, the cytoskeleton. Cytoskeleton is made up of proteins, and there's a number of different types of proteins that are used to produce the structure of the cytoskeleton. And the cytoskeleton has a number of different functions. They can be used to support the cell, they can be used for transport within the cell, and they can be used for the movement of the cell itself. And there's actually three categories of cytoskeleton. You'll see it made up of microfibers, intermediate filaments, and microtubules. Now here I'm just showing more detail about a ribosome and its function. You can see here in red here, this is the messenger RNA that's being read. So the ribosome will, and there's two parts of it. It assembles in the presence of a messenger RNA. So these two pieces are normally floating independently, but they come together when there's a messenger RNA. And then they'll actually move along the messenger RNA. And the messenger RNA has the code in it. So you remember you have adenine, right? Adenine, guanine, cytosine, and uracil that makes up messenger RNA. The ribosome will read those base pairs, and each that actually encodes for amino acids. So you can see a protein that's coming out of the bottom here. This protein is actually being assembled. It's made of amino acids. The amino acids are assembled, and you're making this protein. And this ribosome could be on the surface of the rough endoplasmic reticulum, or it could be free floating in the cytoplasm of the cell. If the ribosome is producing a protein, the two pieces are together. When it's not producing protein, when it's not reading a messenger RNA, the two independent pieces, the two parts of it actually are floating independent.